Okay, so let's jump right in. Imagine having a blueprint for every single tiny machine that makes life possible, but you have no idea what any of them actually look like. I mean, for 50 years, that was the reality for biologists. Today, we're diving deep into AlphaFold, the revolutionary AI that finally solved this massive challenge and, honestly, changed biology forever.
Right, so let's get to the very heart of it. For over half a century, this wasn't just some academic puzzle. No, this was a fundamental roadblock. You see, understanding how proteins fold is the key to everything. Understanding diseases, creating new medicines, even designing new biological materials. It was the bottleneck holding back a whole universe of scientific progress.
So, what is this problem really? Think of it like this. Imagine I hand you a long list of car parts, every single nut, bolt, and panel. Now, just from that list, I want you to predict the exact complex three-dimensional shape of the fully assembled sports car. That's pretty much the protein folding problem. You've got the blueprint, the amino acid sequence, but you need to know what the final working machine actually looks like. So here's how we're going to break it all down. First, we'll really get a feel for the massive scale of this 50-year challenge. Then, we'll unpack AlphaFold's completely new approach and take a peek inside its neural network. After that, we'll look at the absolutely mind-blowing accuracy it achieved. And finally, we'll explore how this is already changing the future of science.
You know, for decades, our ability to read the blueprints of life just completely outpaced our ability to understand what they meant. We were basically drowning in data, but totally starved for real insight.
And this right here really puts the problem into perspective. On one side, thanks to genomic sequencing, we had this mountain of data, billions of protein sequences, billions. But on the other side, figuring out the 3D structure of just one of those proteins, well, that was a brutal, grueling process, often taking years of lab work. The gap between what we knew and what we understood was just getting wider every single day.
And that brings us to the breakthrough. DeepMind's AlphaFold. It didn't just inch forward. It completely forged a new path, totally redefining what was even possible with computers.
Okay, this is where the magic really happens. For decades, scientists were kind of stuck in two different camps. One group tried to solve the problem with pure physics, like a super complex geometry puzzle. The other group looked for clues in evolutionary history, comparing similar proteins across different species. AlphaFold's absolute genius was in realizing it wasn't an either-or question. It fused both approaches, creating an AI that understood not just the physical laws of how atoms interact, but also the biological patterns that evolution carved out over millions of years.
So how does this thing actually work? Let's peel back the layers a bit and get a high-level look at the mechanics of this incredible neural network. You can really think of the process in three main stages. First up, the input. AlphaFold takes the protein sequence and compares it to a massive database of known proteins. This gives it what's called a multiple sequence alignment, or MSA, which is basically a collection of evolutionary relatives that provide clues about which parts are important. Second, the processing. This is where these Evoformer blocks come in. They analyze both the physical constraints and that evolutionary data to create a kind of relationship map of the protein. And finally, the output. A structure module takes that map and uses it to build the final, precise 3D model, literally atom by atom.
Now, what makes AlphaFold so powerful isn't just one single trick. It's more like a symphony of innovations. The Evoformer blocks are designed to think about evolutionary data and physical space at the same time. But the real game changer? It's this iterative process. The model makes an initial guess, then it feeds that guess back into itself to refine it, getting more and more accurate with each loop. And here's a wild twist. It also uses a technique called self-distillation. It makes predictions on proteins where we don't know the structure. And if it's really confident in a prediction, it just treats it as a new piece of training data. I mean, the AI literally teaches itself how to get smarter.
This is where the rubber meets the road. All these clever ideas, they sound great, right? But how do we know they actually work? Well, we look at its performance in CASP-14. Think of it as the Olympics of protein structure prediction. The results were, well, to put it mildly, they were staggering. This chart shows the backbone accuracy, basically how well the prediction matches the main structural scaffolding of the protein. The measurement here is angstroms. We'll get to that in a sec. And lower is better. As you can see, AlphaFold hit an accuracy of 0.96. The next best method? It wasn't even close, coming in at 2.8. This wasn't just a small step forward. This was a complete game changer.
Let's really let that number sink in for a moment. 0.96 angstroms. What does that even mean? Well, an angstrom is a ridiculously tiny unit of length, and a single carbon atom is about 1.4 angstroms wide.
So this means AlphaFold was predicting the structure of these complex biological machines with an error margin smaller than the width of a single atom. That is a level of accuracy that before this you could only get through years of incredibly difficult and expensive lab experiments.
And get this, it wasn't just about the general shape of the protein's backbone. When you look at the placement of every single atom, including all the complex little side chains, AlphaFold's lead was just as huge. It hit an all-atom accuracy of 1.5 angstroms, more than twice as good as the runner-up. It was a total, undeniable clean sweep.
So, okay, we have this incredible atom-level accuracy. What does that actually mean for the real world of science and medicine? And maybe just as importantly, what can't it do? At least, not yet.
The potential here is almost hard to wrap your head around. It can speed up drug discovery by modeling a disease's target protein in minutes instead of months. It lets scientists design new enzymes for everything from manufacturing to cleaning up the environment, and it gives us immediate insight into genetic diseases, helping us see how one tiny change in a sequence can lead to a devastating change in structure and function. It's a massive accelerator for all of biological research.
But, and this is important, it's not magic. AlphaFold has very clear boundaries. Its accuracy really depends on having enough evolutionary data. It needs that multiple sequence alignment to have at least 30 or so sequences to do its best work. It also has a tough time predicting how multiple different protein chains interact with each other, and yeah, it still requires a serious amount of computational power to run.
You know, in the end, AlphaFold's biggest contribution wasn't just answering a 50-year-old question, it was giving us an entirely new language to speak with biology. By turning the 1D blueprint of life into a 3D functional reality, it has opened up a whole new universe of questions and possibilities, and it's going to fundamentally accelerate the pace of discovery for generations to come.